Good evening, everyone. I welcome you all to the weekly live session on the NPTEL Bioinformatics Algorithms and Application course. So in this, I'll discuss the summary of the weekly lectures, followed by some sample problems along with the questions of the portal. So to start with, here I wanted to start today with protein folding. Protein folding is a process by which a polypeptide chain folds to become a biological active protein in its native 3D structure. Protein structure is crucial to its function. Folded proteins are held together by various molecular interactions. During translation, each protein is synthesized as a linear chain of amino acids or a random coil which does not have a stable 3D structure. During translation, each protein is synthesized as a linear chain of amino acids or a random coil which does not have a stable 3D structure. The amino acids in the chain eventually interact with each other to form a well-defined sugar protein. The amino acid sequence of a protein determines 3D structure. Holding of a protein into the correct native structure is a key to their function as shape defines the function. Failure to fold properly produces inactive or toxic proteins that malfunction and causing numerous diseases. There are four stages of protein folding. The folding of a protein is a complex process involving four stages that gives us the various 3D protein structures essential for diverse functions. In the human body, structure of a protein is hierarchically arranged from a primary sequence to quaternary sequence as shown in each. The wide variation in amino acid sequence accounts for different conformations in protein structure. Primary structure refers to the linear sequence of amino acid residues in the polypeptide chain. Secondary structure is generated by the formation of hydrogen bonds between the atoms in polypeptide backbone, which folds the chain into either alpha helices or beta sheets. We have tertiary structure, which is formed by the folding of secondary structure sheets or helices into one another. This tertiary structure of a protein is a geometric shape of a protein. It usually has a polypeptide chain as a backbone with one or more secondary structures. And this tertiary structure is also determined by the interactions and bonding of amino acid side chains in the protein. Finally, we have quaternary structure, which results from a folded amino acid chain to tertiary structures interacting further with each other to give rise to a functional protein such as hemoglobin or DNA polymerase. Factors affecting protein folding. Protein folding is a very sensitive process that is influenced by several external factors including electric and magnetic field, temperature, pH, chemicals, space limitation, and molecular crowding. These factors influence the ability of the protein to fold into the correct functional form. Extreme temperature affects the stability of the proteins and causes them to unfold or denature. Similarly, extreme pH, mechanical forces, and chemical denaturants can denature proteins. During denaturation, protein lose their tertiary and secondary structures and become a random coil. Although denaturation is not always reversible, some proteins can refold under certain conditions. Some cells contain heat shock proteins or chaperones that protein protects in the cell against heat denaturation. These chaperones help proteins to fold and remain under extreme temperature. They also assist misfolded proteins in unfolding and refolding correctly. So now I have a question. What factor can influence the ability of a protein to fold into the correct functional form? Whether cofactors or chemicals, temperature and pH, or molecular counting, or all of the above. So the question is about what factors can influence the ability of a protein to fold into the correct functional form. As we discussed, the answer is all of the above. Protein folding can be influenced by various external factors, including temperature, pH, chemicals, and environmental conditions such as space limitation and molecular crowding. However, what happens to proteins during denaturation? They gain their tertiary and secondary structure, they lose their tertiary and secondary structure, they remain in their native 3D structure, or they become more stable. What happens to protein during denaturation? Whether they have a gain in structure or loss in their structure, or remain as it is, or become more stable. Yes. So in generally, denaturation is defined as a loss in their tertiary and secondary structure. So because denaturation disturbs the native 3D structure of a protein, causing them to fold and lose their functional conformation. So because of these denaturation or misfolding, some diseases tend to occur. So here now I'll be discussing about diseases related to incorrect protein folding. So misfolded proteins denature easily and lose their structure and function. Incorrected proteins can lead to, incorrect protein folding can lead to many human diseases. Alzheimer's disease is an example of neurodegenerative conditions caused by protein misfolding. This disease is characterized by the dense plaques in brains caused by the misfolding of secondary beta sheets. Secondary beta sheets of liberal beta amyloid proteins present in brain matter. Huntington's disease and Parkinson's disease are other examples of neurodegenerative diseases associated with protein misfolding. Cystic fibrosis is a fatal disease caused by misfolding of the CFTR protein, which is cystic fibrosis transmembrane conductance regulator. In most cases of cystic fibrosis, the phenylalanine at position 508 of CFTR is deleted, causing misfolding of the regulator protein. Several allergies have also been shown to be caused by incorrect protein folding. The protein can misfunction for several reasons. When a protein is misfolded, it can lead to denaturation of a protein. Denaturation is the loss of protein structure and function, and the misfolded does not always lead to complete lack of function, but only partial loss of functionality. The misfunctioning of proteins can sometimes lead to diseases in the human body. One such is Alzheimer's disease. So Alzheimer's is a neurological degenerative disease that affects around 5 million Americans, including half of those who are age 25 or above. The predominant risk factors of Alzheimer's are age, family history, and heredity. Alzheimer's disease typically results in memory loss, confusion of time and space, misplacing places, and changes in mood and behavior. Alzheimer's also results in dense plaques in the brain that are comprised of nuclear beta amiloid proteins with a well-ordered beta-sheet secondary structure. These plaques usually look like voids in the brain matter and are directly connected to the deterioration of the thought processes. It has been determined that AD is a protein misfolding disease, where the misfolded protein is directly related to the formation of these plaques in the brain. It is yet to be fully understood that what exactly causes this protein misfolding to disease, but several theories point out to oxidative stress in the brain to be the initiating factor. This oxidation results in the damage to the phospholipids in the brain, which have been found to result in a faster accumulation of amyloid beta protein. As I discussed, we also have cystic fibrosis, which is a chronic disease that affects more than 30,000 American population. So this here the misfolding happens in CFTR protein, which can result in the potentially fatal disease. In approximately 70% of the cystic fibrosis cases, a deletion of phenylalanine at position 508 in CFTR is observed. This deletion of phenylalanine at 508 seems to be directly connected to the formation of cystic fibrosis. The field of computational studies of protein folding encompasses various methods and approaches aimed at understanding the complex processes by which proteins assume their functional three-dimensional structures. One of the fundamental concepts in this field is Levinthal's paradise, proposed by Cyrus Levinthal in 1868. Levinthal observed that the number of possible conformations for an unfolded polypeptide chain is astronomically large due to the numerous degrees of freedom involved. If a protein were to fold by sequentially sampling all the possible conformations, it would take an impractically long time, much longer than what it is observed experimentally. This paradox led to the revelation that proteins must fold through a series of metastable intermediate states rather than randomly sampling conformations. The energy landscape of protein folding provides a conceptual framework for understanding how protein navigates their folding path. According to this view, proteins fold into their native structures through a funnel-shaped energy landscape where the native state presents the lowest energy state. This funnel-shaped landscape allows protein to explore various folding pathways and intermediates while ultimately converging to native state. The theory of minimal frustration suggests that the natural selection has optimized the protein sequences to minimize the frustration of folding processes, ensuring efficient folding kinetics and stability. Computational modeling plays a crucial role in studying protein folding. Techniques such as molecular dynamic simulation allow researchers to simulate the protein folding process and explore the dynamics of the protein in silico. These simulations can provide insights into the protein folding mechanisms, kinetics, and structural stability. However, due to computational limitations, simulation of larger proteins or long-term folding process remains challenging. There are several large-scale computational products aimed to crowdsource computing power for protein folding simulations. These projects contribute to our understanding of protein folding and facilitate the development of new computational methods. In recent years, advances in artificial intelligence have led to breakthroughs in protein structure predictions. DeepMind's AlphaFold, for example, achieved a remarkable accuracy in predicting protein structures as demonstrated by its performance in CASP competition, that is critical assessment of structure prediction competition. AlphaFold structures represents a significant advancement in computational biology and brings us closer to solving the long-standing challenge of predicting protein structures accurately. While AlphaFold's achievements are transformative, challenges remain, particularly in understanding the underlying rules and mechanisms of protein folding. Nevertheless, these advances highlight the immense potential of computational methods in elucidating the complex process underlying the protein folding and structure prediction. So here I have a set of questions. We'll try to answer them one by one. So what is the structure of a protein that refers to the local folding of polypeptide chain? What refers to the structure of a protein that refers to the local folding of a polypeptide chain? What refers to the structure of a protein with local folding of the polypeptide chain? Okay, we will be seeing answers all at once. So it is secondary structure of a protein that refers to the local folding of a polypeptide chain. Then second one, so what structure of a protein refers to the overall 3D arrangement of its secondary structural elements? So the overall 3D arrangement of the secondary structural elements in a protein is referred to as what level of structure? So this is tertiary structure of a protein that refers to the overall 3D arrangement. Then I have the third one, so which is what structure of a protein refers to the association of multiple protein subunits to form a functional complex. This association of multiple protein subunits to form a functional. So it is the quaternary structure of a protein refers to the association of multiple protein subunits to form a functional complex. Then we have the fourth one. Hydrogen bonds between amino acid residues are important for formation of what level of the structure? So hydrogen bonds drive the formation of what level of the protein structure, whether it's a primary, secondary, tertiary, or quaternary. So hydrogen bonds are important for the formation of what level of protein structure. So generally hydrogen bonds are formed to stabilize the protein structure such as to form the protein structure such as alpha recess and beta sheet. So it is secondary structure. Or structure of a protein can be disrupted by denaturing such as heat or extreme pH. It is generally tertiary structure. So last one is a pretty simple and straightforward. The alpha helix and beta sheets are examples of secondary structure of a protein. So here I have provided answers. Kindly go through these answers in the questions and try to match with your answers. I'll wait for a couple of minutes here. Okay, moving forward to the topic of protein folding rate. So protein folding rate refers to the speed at which the protein molecule adopts its native three-dimensional structure from a random unfolded state. The process of protein folding involves integrate movements and interactions between different parts of the protein molecule, leading to the formation of stable secondary and tertiary structures. Several factors influence the protein folding rate of a protein. So they are size and complexity, primary structure, environment, denaturation and chemicals, and energy landscape. Generally, smaller proteins tend to fold more quickly than larger ones due to the fewer number of possible conformations they can adopt. However, the folding rate also depends on complexity of protein structure and number of interactions needed for proper folding. Then we have primary structure. So the sequence of amino acids in protein's primary structure determines the folding pathway and influences the folding rate. Some amino acid sequences naturally promote rapid folding, while others may lead to misfolding or aggregation and slowing down the process. Then we have environment, the surrounding environment, including factors such as temperature, pH, ionic strength, and presence of other molecules, can significantly affect the folding rate. Optimal conditions for protein folding may vary depending on specific protein. Then we have chaperone proteins. Molecular chaperones assist in protein folding by preventing misfolding and aiding in the correct folding pathway. These chaperones can enhance the folding rate by stabilizing intermediate structures and preventing aggregation. Then we have denaturants. Certain denaturant agents or chemicals can disrupt the protein structure and slow down the folding process. Conversely, some additives or stabilizing agents may accelerate folding by providing favorable conditions or stabilizing intermediates. Protein folding occurs through a complex energy landscape where the protein searches for its native state while navigating various intermediate states. The folding rate depends on the energy barriers between these states and probability of transitioning between them. Understanding protein folding rates is crucial for studying protein structure, function, relationship, as well as for applications such as drug design and biotechnology. Various experimental and computational techniques such as kinetic studies, molecular dynamic simulations, and bioinformatic analysis are employed to investigate and predict the protein folding rates under different conditions. So here now I have a question. So according to the energy landscape model, what represents the lowest energy state for a protein? So the state with the lowest energy state is called as a native state or denatured state, intermediate state, or the unfolded state. The state with the lowest energy is the native state. In the energy landscape model, the native state of a protein represents the lowest energy state towards which the protein folds. How do molecular chaperones influence protein folding? Whether these molecular chaperones induces denaturation or promote protein aggregation or prevent misfolding or aiding in the correct folding pathway or disrupting hydrogen bonds. What do molecular chaperones do or try to influence? In what way they try to influence protein folding? They prevent misfolding and aid in correct folding pathway. Molecular chaperones assist in protein folding by preventing misfolding and guiding the protein along the correct folding pathway. With the recent advancements in what recent advancement in artificial intelligence has shown remarkable accuracy in predicting protein structures, whether it's DeepMind AlphaGo or deep learning for protein structure prediction or DeepMind's AlphaFold or Google's TensorFlow. So what is the recent advancement in artificial intelligence that has shown remarkable accuracy in predicting protein structures? So DeepMind's AlphaFold achieved a remarkable accuracy in predicting protein structures and representing a significant advancement in computational biology. According to Lewinthal's paradox, why it is impractical for proteins to fold by sequentially sampling all possible conformations. So due to lack of computational power, or due to large number of possible conformations, or due to the absence of chaperones, or due to the presence of chemical denaturing. This is because of Lewinthal's paradox addresses the impracticality of the protein folding by sequentially sampling all possible conformations given the vast number of potential conformations. So now we'll start with the uh, models in protein folding. So protein folding is a dynamic process where a polypeptide chain assumes its functional and three-dimensional structure. Understanding the kinetics of this process is crucial for elucidating how proteins achieve their native states and perform their biological functions. The two-state and three-state models are simplified frameworks commonly used to describe protein folding kinetics, each offering insights into different aspects of protein folding. So to start with the two-state model, in the two-state model, protein folding is assumed to occur directly from the unfolded state to the folded state without any intermediate. The folding rate curve represents the rate at which protein transitions from the unfolded to the folded state. This model suggests that folding proceeds through a single and rate-limiting step at the free energy chain, that is delta GF, associated with folding determines the folding rate constant according to the equation. Uh, folding rate equals to unfolding rate constant into exponential of uh, minus delta GF divided by RT, where Ku is the unfolding rate constant, R is the gas constant, and T is the temperature. The value of energy change associated with the folding can be experimentally determined by using such as equilibrium unfolding experiments. Then we have three-state model. The three-state model acknowledges the possibility of intermediate states during protein folding, adding complexity compared to the two-state model. Here, proteins are assumed to fold through one or more intermediates before reaching the final folded state. The folding rate constant in this model is determined by the rates of two key steps, that is, the formation of intermediates Ki and the final folding step. The overall folding rate constant is described by the equation as shown, that is Ki into K dash F divided by Ki plus K dash F. This model suggests that protein folding involves multiple rate limiting steps where the formation of intermediate plays a significant role. Experimental techniques such as kinetic measurements, stop-to-flow spectroscopy, and NMR spectroscopy are used to determine the folding rate constant and understanding the folding pathways of proteins. These models provide simplified descriptions of protein folding kinetics but may not fully capture the complexity of folding in all proteins. For more integrate folding scenarios involving multiple intermediates or non-equilibrium processes, more sophisticated models may be required. The folding rates of the proteins can be influenced by a variety of factors, including the sequence of amino acids that make up the protein. In general, proteins that fold quickly tend to have higher portion of small and hydrophobic amino acids, while proteins that fold slowly tend to have higher proportion of large polar and charged amino acids. So, talking about the fast folding proteins. Fast folding proteins tend to have a compact and stable interstate, which means that the folding process is relatively fast and efficient. In general, fast folding proteins tend to have a high content of hydrophobic amino acids such as alanine, leucine, and isoleucine, which tends to cluster together in the protein's interior. These hydrophobic interactions help to stabilize the protein's interstate and promote transit folding. One example of fast folding protein is within headspace subdomain, which falls into the native state in just few microseconds. The thick within headspace subdomain contains a high proportion of hydrophobic amino acids such as adenine, valine, and leucine, which help to stabilize compact folding state. Then we have slow folding protein. The other hand, slow folding proteins tend to have a more complex folding landscape, which means that folding process is slower and more prone to misfolding. Slow folding proteins tend to have higher content of polar and charged amino acids such as arginine, glutamate, and lysine, which can form complex electrostatic interactions that slow down the folding process. Slow folding proteins can also tend to have a longer and more flexible region, which can adopt a variety of conformations and make the folding process more difficult. One example of slow folding proteins is enzyme adenylate kinase, which takes several seconds to fold into its native state. Adenylate kinase contains higher proportion of polar and charged amino acids such as glutamate, glutamic acid, and lysine, which can form stronger interactions with other molecules and These interactions can slow down the folding process by creating a barrier for the formation of proteins leading state. However, it is important to note that there are many exceptions to these general rules and the following characters of the protein can be influenced by many other factors, including the presence of post-translational modifications and interactions with the other molecules. Therefore, predictably, folding kinetic group protein is solely based on its amino acid sequence is challenging as in summary. The preference of amino acids in fast slow-folding proteins is generally related to their size, hydrophobicity, and probability. Fast-folding proteins tend to have a higher proportion of small and hydrophobic amino acids, while slow-folding proteins tend to have a higher proportion of larger polar or charged amino acids. However, it is important to note that folding the protein is also influenced by many other factors such as temperature, pH, and presence of other molecules in environment. Folding, protein folding and protein folding rates can vary significantly among different protein structural classes. So we have structural classes as discussed in the last class. These all alpha proteins, all beta proteins, alpha beta and alpha plus beta proteins. So all alpha proteins are characterized by the predominant secondary structure consisting of alpha acids. These proteins tend to fold faster compared to the other structural classes. The relatively simple folding pathway of alpha protein is attributing to the presence of stable alpha acyl that can form early in the folding process. When we have all beta proteins have predominant secondary structure composed of beta strands. They typically fold more slowly than all alpha proteins. The folding pathway of all beta proteins is often more complex, involving the formation of partially folded intermediate states before reaching the fully folded state. We have when we have alpha beta or alpha plus beta proteins. Alpha beta proteins contain both alpha and beta strands, while alpha plus beta proteins have non-contiguous regions of alpha and beta strands. These structural factors exhibit folding rates which fall between those of all alpha and all beta proteins. Their folding kinetics are intermediate, reflecting a balance between the simplicity of all alpha proteins and the complexity of all beta proteins. With respect to general strengths, there is a significant variability between each structural class. Individual proteins within the same class may exhibit unique folding kinetics due to the differences in sequence, size, and specific structural features. Factors such as the rate of acid, specific post-translational modifications, and interactions with the ligand or other molecules can further influence folding rates. Studying the folding rates and mechanisms of different protein structural classes is essential for understanding the fundamental principles of protein folding. Insights gained from these studies can inform the development of new methods for predicting and designing protein structures. Understanding protein folding kinetics also has implications for disease research, drug discovery, and biotechnology applications. In summary, there are overarching trends in the folding rates of different protein structural classes. The variability and the complexity inherent in protein folding highlight the need for detailed studies to uncover the underlying principles governing the essential biological process. So I have a question. So which type of amino acids tend to characterize in faster folding proteins? So what type of Amino acids tend to characterize fast-folding proteins. Fast-folding proteins often tend to have higher proportion of small and hydrophobic amino acids. The low-folding proteins tend to have higher proportion of which of these amino acids with a larger polars, small and hydrophobic, charged or polars, or aromatic and bulky. Slow-folding proteins often have higher proportion of large polar amino acids. This is about the protein folding. Then here I have different types of protein interactions. Protein-protein interactions refer to the physical contacts between two or more proteins, leading to the formation of complexes or assemblies that perform various cellular functions. So we have several types of protein-protein interactions. First one is stable interactions. These interactions are long-lasting and essential for structural integrity and function of multi-protein complexes. Stable interactions are often involved in structural scaffolding, cellular organization, and maintaining the architecture of molecular complexes. Examples include interactions between the ribosome, DNA polymerase, or proteasome. Then we have transient interactions. Transient interactions are relatively short-lived and dynamic, occurring only transiently to regulate the specific cellular processes. These interactions play a crucial role in signaling pathways, cellular regulation, and responses to the external stimuli. Examples include interactions between the signaling proteins in pathways, such as MAP kinase signaling, receptor ligand interactions, and interactions between transcription factors, and regulatory proteins. Protein-protein interactions are highly specific and the proteins are selectively binding to their interacting partners through complementary surfaces or binding sites. Protein-protein interactions exhibit varying affinities ranging from a weak to strong interactions depending on the. And protein-protein interactions are dynamic and reversible, allowing for the regulation and modulation of cellular process in response to the changing conditions or signals. So the mechanisms of protein-protein interactions include lock and key, induced fit model, and allosteric regulation. So in lock and key model proposes that proteins have specific complementary shape that fits together like a lock and key, enabling the specific interactions between them. Then we have induced fit model where protein interactions induce conformational changes in one or both interacting proteins, leading to a more favorable binding conformation. Protein-protein interactions can also allosterically regulate the activity of enzymes or other proteins by inducing conformational changes that affect their catalytic or regulatory properties. So there are methods for studying protein-protein interactions. So we have yeast-2 hybrid assay, co-immunoprecipitation, surface plasmon resonance, and mass spectroscopy. In yeast-2 hybrid assay uses a yeast reporter system to detect the interactions between proteins by measuring the transcriptional activation of reporter genes. Then we have co-immunoprecipitation method, which involves co-precipitating of a protein of interest along with its interacting partners using a specific antibodies, followed by identification of interacting proteins. Then we have surface plasmon resonance, which measures the changes in refractive index of the surface bound proteins in response to the binding interactions with its partner protein, allowing for real-time analysis of binding kinetics and affinity. Then we have mass spectroscopy, which can identify protein-protein interactions by analyzing protein complexes isolated from cells or tissues. So protein-protein interaction plays crucial role in virtually all aspects of cellular function, including signal transduction, gene expression, metabolism, and cell cycle regulation. Understanding the mechanism and dynamics of these interactions is essential for elucidating the molecular basis of cellular processes and disease, as well as for the development of therapeutic inventions targeting protein-protein interactions. Then we have protein nucleic acid interactions involving the binding of proteins to specific regions or sequences within DNA or RNA molecules, resulting in the formation of protein DNA or protein RNA complexes that perform diverse biological functions. So we have two types, that is sequence specific and sequence non-specific protein nucleic acid interactions. In sequence specific interactions, protein recognize and bind to the specific nucleotide sequences within DNA or RNA molecule. These interactions are characterized by the high specificity and affinity, allowing proteins to regulate gene expression, DNA replication, and RNA processing with precision. Non-specific interactions occur when proteins bind to the nucleic acids in a more general or non-specific manner without strict sequence specificity. These interactions typically involve electrostatic interactions and play crucial roles in processes such as chromatin organization and nucleic acid packing. So these proteins can recognize the specific DNA sequences by interacting with the major or minor grooves of the DNA double helix, using amino acid side chains to form hydrogen bonds or other interactions with exposed bases. Proteins containing HTF, that is helix turn helix motif, such as transcription factors, can insert specific DNA binding helices into the major groove of DNA, allowing for the sequence specific recognition and binding. Then we have zinc finger proteins, which uses zinc ions to coordinate specific amino acid residues, forming a finger-like structure that recognizes and binds to the specific DNA sequences in a sequence-specific manner. So we have several methods for studying the protein nucleic acid interactions. EMSA, which is electrophoretic mobility shift assay. So this technique involves incubating a labeled nucleic acid probe with the protein extracts and separating protein bound and unbound nucleic acids by gel electrophoresis to detect protein DNA, protein RNA complexes. Then we have chromatin immunoprecipitation. So this allows for the identification of in vivo protein DNA interactions by cross-linking proteins to the DNA in intact cells, followed by immunoprecipitation of the protein of interest and analysis of associated DNA sequences. Then we have crystallography or NMR spectroscopy. These structural biology techniques can elucidate the atomic details of protein nucleic acid complexes, providing insights into the mechanism of recognition and binding. So these protein nucleic acid interactions play an essential role in regulating gene expression, DNA replication, RNA processing, and other cellular processes. Understanding the mechanism and dynamics of these interactions is crucial for deciphering the molecular basis of genetic regulation for developing therapies targeting nucleic acid protein interactions in diseases such as cancer and genetic disorders. So other than that, we have protein lipid interactions and protein small molecule interactions. Just protein lipid interactions involve physical contacts between proteins and lipids crucial for various cellular processes such as membrane trafficking, signaling, and maintaining membrane structure. So we have different types of protein lipid interactions, electrostatic interactions and hydrophobic interactions. Electrostatic interactions occur between charged amino acid residues on the protein and charged lipid head groups in the lipid bilayer. Electrostatic interactions contribute to the association of peripheral membrane proteins with cell membranes, and hydrophobic regions of proteins interact with the hydrophobic tails of lipid molecules, stabilizing protein lipid complexes within the lipid bilayer. These interactions are essential for insertion and anchoring of integral membrane proteins. So protein lipid interactions can be specific, with protein selectively binding to the lipid species or lipid microdomains within the membrane. Protein lipid interactions influence membrane dynamics and organization, affecting processes such as membrane curvature and lipid raft formation. And protein lipid interactions play a crucial role in mediating cellular pathways, membrane trafficking, and protein localization within the membranes. So, liposome binding assay can be used for studying protein lipid interactions. Liposome composed of a defined lipid compositions can be used to assess the binding of a protein to specific lipid species or membrane mimetics. Or other than that, fluorescently lipids and proteins can be visualized using fluorescence microscopy to study their localization or dynamics within the cellular membranes. Then also we have biolayer interferometry or surface plasma resonance. Finally we have small molecule interactions. So we have regulatory interactions. In regulatory interactions, small molecules bind to the specific site on proteins, modulating their activity or function. These interactions can either activate or inhibit protein function depending on the nature of the small molecule and its binding site on protein. Examples include enzyme activation by allosteric regulations or enzyme inhibition by drugs. The structural interactions involve the incorporation of small molecules into protein structure, where they contribute to protein stability or function. This may also involve the binding of cofactors or prosthetic groups essential for protein activity, or the binding of ligand that induces conformational change. include the binding of heme to hemoglobin for oxygen transport and binding of retina to rhodopsin inhibition. So these small molecules interactions can also be studied by X-ray crystallography, NMR, SPR, biolayer interferometry, fluorescent based arrays and computational modeling. So this is what. So I have a question here. So the answers are provided. So that's given a protein, tertiary structure determines its ability to interact with other proteins. Proteins that binds to DNA regulate the gene expression are known as transcription factors. The process by which a protein loses its shape and function due to exposure to extreme conditions such as heat or pH is called denaturation. A protein's binding site determines its affinity for particular ligand. Proteins can interact with each other to physical contacts such as hydrogen bonds and van der Waals interaction. Yeah, kindly go through the answers once. I'll wait for a minute here and then proceed to how to recognize computationally protein binding sites. Amino acids exhibit different binding propensities depending on the type of biomolecular interactions such as protein-protein, protein DNA or protein ligand interactions. These propensities are influenced by various factors including the chemical properties of amino acids, the structure of the interacting molecules and the specific requirements of the interaction. So understanding these binding preferences is crucial for designing effective drugs and therapies targeting specific interaction. So first method to predict these is binding site prediction is a key aspect of paternalizing interactions and drug discovery. Several computational methods have been developed to predict the binding sites of proteins including distance-based, accessible surface area-based and energy-based method. So distance-based methods predict the binding sites based on the spatial proximity of protein residues to a known ligand binding site or ligand molecules. They identify the regions on protein surface that are within a certain distance threshold from ligands are known as binding sites. So these algorithms are relatively fast and computationally efficient and suitable for identifying pockets or cavities on protein surfaces. Sometimes may miss binding sites that are not located within the well-defined pockets or cavities. Limited to predict the binding sites in flexible regions of proteins. Then we have accessible surface area based methods, so which predict the binding sites based on the solvent accessible surface area of protein residues. Regions of the protein surface with higher accessible surface area are more likely to interact with ligands. So this provides a comprehensive view of protein structure and captures potential binding sites beyond predefined pockets or cavities, but limited to the limited ability to predict the binding affinity of ligands and may require additional analysis to prioritize the potential binding sites. Then we have energy based method energy based methods predict the binding sites based on the free energy of interactions between protein and ligand. They typically involve computational docking or molecular dynamic simulations to calculate the binding energy and identify favorable binding sites. So the choice of the method depends upon various factors such as the research question, availability of computational resources, and characteristics of protein ligand system being studied. Combining multiple methods for or integrating experimental data can enhance the accuracy of binding site prediction. In summary, each method for predicting the binding sites has its own strength and limitation, and researchers can offer choose a method or a combination of different methods based on their requirement. Now, we'll study the preference of. So this shows the preference of amino acids across. So this will come after this quiz session. So protein, first question in the quiz today is protein folding rates provide the information on what? Whether it provide the information on 3D structure of the sequence, or how fast or slow a protein can fold, how it assumes its native structure, or all of the above. So protein folding rates tells how fast it assumes its native structure. So we have a concept of chevron plots. So chevron plots are graphical representations used in the study of protein folding kinetics. They are named after the characteristic V-shaped pattern observed when plotting the rate of a reaction against the concentration of denaturant. These plots are particularly useful in elucidating the mechanism and energetics of protein folding transitions, especially for two-state proteins. So chevron plot is typically constructed and interpreted. So protein folding kinetics are monitored using techniques such as top-to-flow or continuous flow, rapid mixing rapid mixing technique combined with spectroscopy techniques like fluorescence or CD. The protein of interest is subjected to increasing concentration of denaturants or varying temperature, and the rate of folding or unfolding reactions is measured at each condition. On the x-axis, the concentration of denaturant, or on the y-axis, the observed rate constant for folding is used. For a two-state protein folding, the chevron plot typically exhibits a characteristic V shape reflecting the transition between folded and unfolded state. The rising of the limb V represents the folding arm, where the increasing denaturant concentration or temperature promote the rate of protein folding, and the falling limb of V represents the unfolding arm, where the increasing denaturant concentration or temperature accelerate the rate of protein unfolding. The point where the folding and unfolding arms intersect is known as the folding midpoint or the transition state, which corresponds to the conditions where the folding and unfolding rates are equal. So the slope of the folding arm provides insights into the rate limiting step of a protein folding under native conditions. Similarly, the fold of unfolding arm indicates the rate limiting step of protein unfolding. So deviations from ideal chevron plot may suggest the presence of intermediate states or non-two-state folding behavior. So here I have a question. Chevron plot is used for to understand protein folding kinetics or thermodynamics of protein folding or predict 3D structures or predict function. So used for understanding protein folding kinetics. Which database contains the binding affinity of protein-protein complexes upon mutations? So proximate. For binding site residues, PDB param provides information on distance between the residue pairs. So yeah, this is what I have for today. So I have left one topic is protein interactions. So amino acids exhibit different binding propensities depending on type of biomolecular interactions such as protein-protein, protein-DNA or protein-ligand interactions. These propensities are influenced by various factors including chemical properties of amino acids, structure of interacting molecules, and the specific requirements of interactions. Understanding these binding preferences is crucial for designing effective drugs, therapeutic therapies, and targeting specific interactions. So, in protein-protein interactions, hydrophobic residues are commonly found in protein-protein interfaces due to their ability to form hydrophobic reactions with the other hydrophobic residues. They stabilize protein complexes by minimizing the exposure to the surrounding aqueous environment. Aromatic amino acids frequently found in protein-protein interfaces are capable of forming paper stacking interactions and hydrogen bonds, contributing to the protein stability and specificity. Charged amino acids are important for protein-protein interactions as they can form salt bridges and electrostatic interactions, contributing to the specificity and strength of binding between proteins. Then we have protein DNA interactions. Basic amino acids are commonly found in DNA binding proteins due to their ability to form electrostatic interactions with a negatively charged phosphate backbone of DNA, facilitating protein DNA recognition and binding. Aromatic amino acids can contribute to the protein DNA interactions by forming pi-pi stacking interactions with DNA base pairs and stabilizing protein DNA complexes. And certain polar amino acids also participate in hydrogen bonding with DNA bases, enhancing specificity in protein DNA recognition. Then we have protein ligand interactions. So hydrophobic amino acids predominantly found in ligand binding pockets, facilitating hydrophobic interactions with the ligand. They contribute to the ligand recognition and binding affinity. And we have aromatic amino acids also, which can form pi-pi stacking interactions with the ligands, enhancing the binding affinity and specificity in protein ligand interactions. And we have polar amino acids, which are capable of forming hydrogen bonds with ligands, contributing to ligand recognition and specificity. Then we have charged amino acids important for forming salt bridges and electrostatic interactions with charged ligands, influencing binding affinity and specificity. So the relationship between the contact order and long range order and the folding rate is crucial in understanding protein folding kinetics. Contact order refers to the average sequence separation between residues that form local contacts in a folded state of a protein. Proteins with low contact order tend to this is because low contact order proteins have more compact and stable native state. Long range order, on the other hand, refers to the degree of correlation between amino acid interactions in the folded state of a protein. Proteins with high long range order tend to fold more rapidly than proteins with low long range order. This is because long range order proteins have high long range order proteins have more cooperative folding process where the formation of one long range interactions helps to stabilize the formation of other long range interactions. Now considering both the factors together, proteins with low contact order or the high long range order fold the fastest. These proteins have a compact native state that requires fewer long range interactions to stabilize, and interactions that do occur are highly coordinated. And conversely, proteins with high contact order and low long range order fold the slowest. These proteins have a more extended native state that necessitate the numerous long-range interactions for stabilization. Additionally, the lack of coordination in these interactions may impede the folding process, leading to slower kinetics. In summary, the folding rate of a protein is influenced by both contact order and long range order, with proteins sorry proteins exhibiting low contact order and high long range order folding the fastest, and those with high contact order. So to let's clarify the relationship between the contact order and long range order. Proteins with higher contact order have a greater average sequence separation between the residues that form non-local long-range contacts in the folded protein. This implies that these proteins have more long-range interactions to form. Proteins with higher contact order tend to fold more slowly. This is because the process of forming numerous long range interactions requires more time and typically more complex. The extensive rearrangements needed to re-establish these long range contacts contribute to a slower overall folding rate. Proteins with higher long range order exhibit a greater degree of correlation between amino acid interactions in the folded state. This is because while these proteins have coordinated interactions, the process of establishing these interactions can be slower. So the cooperative nature of long range interactions may require sequential stabilization of multiple regions of protein, leading to a overall folding kinetics. In summary, while proteins with higher contact order indeed fold more slowly due to complexity of forming numerous long range interactions, proteins with a higher long range order also tend to fold slowly because of the time consuming process of establishing coordinated interactions across the protein. Therefore, both factors contributing to slower folding end rates and albeit through the different mechanism. So that's all for today. Yeah, any other questions I can take? If no other questions, you are free to leave. I'll stay in this call for some more time.